Thank you, John. Hello, everyone. Good evening. First of all, thanks, John and the CSR community for inviting me here to talk about the human microbiome. So for this particular talk, I sort of divide it into two main parts. So for the first half of the talk, I'm going to try and give a bit of a broad overview of the human microbiome field. What's the current state of the art? What do we know about the human microbiome? There's a lot of misconception, a lot of hyperbole. I'm sure lots of you have heard about the human microbiome in different sources in the media, on TV. And then in the second half of the talk, I'm going to focus a bit more on what the research we are doing is revealing. So as John mentioned, so I'm based at the Department of Veterinary Medicine. I lead the Microbiome Function and Diversity Group. And our specialty is really in using DNA sequencing approaches, the methods we call metagenomics, to really try to understand what is the relevance of the human microbiome for health. Now, but first, to get things started and to warm things up a bit, I have prepared here a short quiz for those in the audience. Apologies for those that are online. I've prepared 3 short statements, true or false. I'm just wondering to gauge kind of what's everyone's opinion on this. So there are more stars in the Milky Way galaxy than microbes in our gut. Who says true? Okay, we have a few. Who says false? Nice, okay, good job. Milky Way, yes, it's 10 times different. So we have about 30 to 40 trillion microbes in our gut, whereas the current estimates in terms of number of stars is much higher than that. So yeah, good job. Second one, we have 10 times more microbes than human cells in our body. Who says true? Okay, quite a few. Who says false? Oh, interesting. It's actually false. So this is one of the sort of misconceptions that sometimes we hear. This 10 times number is actually thrown around quite a bit, but there was a paper a few years ago that actually tried to have a more careful estimate of the difference, and they realized we're essentially as much microbes as we are human. The numbers are still a bit higher on the microbiome side, so they still win, but it's not as much of a difference. Lastly, our gut microbiome weighs 1 to 2 kilograms. True or false? Who says true? Okay. Who says false? Okay, good job, good job. Okay, so we got two out of 3. That was a pretty good score. Yes, that's one of the other figures that actually is used quite a lot in terms of the weight, but actually in certain studies have used post-mortem samples. They actually realize that colonic contents only represent about 100 to 400 grams. And this includes fecal material from the actual host, sort of like epithelial cells. So the gut microbiome is actually probably going to be a small portion of that. Okay, so yeah, so we do have quite a lot of microbes in and on our body. So as I mentioned before, we have about 40 trillion microbes that exist all around us, and the majority of them actually exist in our intestinal tract. In terms of the human cells, we're about 30 trillion human cells. So again, yeah, we're as much microbe as we are human. What's interesting, though, is that if you think at a genetic level, each one of us, we're pretty much more than 99.9% genetically identical. But our microbiome actually varies quite a bit. So we can see inter-individual differences going beyond 80%. So if you think everybody here in this room, we probably only share about 20 to 30% of our microbiomes. And this is at the species level. If you go deeper to higher resolutions, those individual strains and lineages, this number is much higher. So try to understand what drives that variation and how that variation affects health and disease has been one of the main challenges in the field. And the microbiome field has actually evolved quite a bit. Although we think that the microbiome field is relatively recent because all the news that had come out and all the use of these techniques to understand the microbiome in health, we actually can trace back the history of the field quite early into the 20th century. So obviously at the time, we were using much more rudimentary techniques, but there were certain developments that actually made the difference, that actually sort of sparked the beginning of this microbiome field. One of the big things was about the use of methods to actually be able to grow the bacteria that are sensitive to oxygen exposure, so these called anaerobes, so using specific what are called Hungate tubes to be able to grow bacteria in the absence of oxygen. The use of germ-free mice, so mice that are depleted of all microbes that I can actually then test and see what impact specific microbes have. The use of fecal transplants, yes, this is as gross as it sounds, where essentially you are transplanting the fecal material from one individual to another. and other things like looking at microbiota in early life. But honestly, the big revolution in microbiome science really came with the development of DNA sequencing. And this is really when the field took off, when we're able now to use DNA sequencing approaches to profile the microbiome, understand what species were there, and actually differentiate individuals in different contexts. So in a typical microbiome study where these technologies are used, let's imagine you want to understand, for instance, whether a microbiome is linked to a specific disease. What usually happens is you have your particular population of interest of the disease you're looking at, so for instance, your case group, and then you have a control population, so this would be your healthy group. What researchers usually do is they look at the microbiome composition of these two populations. So here you can think of these different colored shapes as different bacteria. They try to identify which species are associated with one group versus another by actually quantifying and measuring all these different species. So in this particular instance, you could assume, for instance, that the bacteria that are in blue are associated with disease. And this is how the field has actually done a lot of the research that we know to date. So what we know now is that there's strong associations between the composition of the microbiome and between specific species with numerous diseases. So we have things like colorectal cancer, inflammatory bowel disease, obesity, that are strongly linked to specific combinations of species that are found in these populations. But there's newer research coming out that actually is being even more ambitious about linking the microbiome with things you might not naturally expect. Things like neurological disorders. So there's a huge wave of research now, what is called the gut-brain axis. There are things like physical fitness, behavior, where people are really trying to understand how far does the microbiome actually affect our health and our behavior. Having said that, there are substantial issues with a lot of research that is done on the microbiome. And one thing that to keep in mind is that depending on the methods, depending on the techniques that researchers use, you can actually have very different results. And I'm highlighting here two papers that illustrate how the use of different techniques and the lack of awareness of certain limitations of the microbiome field can lead to substantial problems. So the paper here on the left essentially shows how the use of microbiome samples coming from low biomass sites, so these are essentially body sites that have a low level of microbes, for instance, our skin or the respiratory tract, can essentially inflate certain signals coming from contamination. So you imagine that when people are processing these samples in the lab, there is actually bacterial DNA present in all the reagents and all the kits that people use to process these samples. So if you don't have a lot of material to begin with, a lot of this contaminated signal comes out. So there's a lot of papers talking about, for instance, the placenta microbiome. This has also been a huge controversy in the field, when in reality, the placenta is supposed to be sterile. So current research actually points that there are no microbes, that the infant is only colonized by the microbiome after birth. So that means a lot of the research has been done looking at what is called the placental microbiome is actually looking at contaminated reagents and kits. Another paper that I've highlighted here on the right was a paper that originally tried to show that the use of microbiome profiling in blood and in cancer tissues could be used as a diagnostic approach to identify when an individual was developing cancer as a form of early prevention. But what actually came out is that the use of the techniques and the sample processing that researchers use actually, again, led to erroneous signal that actually completely confounded the results. And this actually led to a retraction of this paper last year. So the microbiome is a really complex area that requires very careful thought about the techniques and the analysis that are being done at the risk of potentially doing inappropriate things. So the field has really gone through this sort of hype cycle, I feel, in the last few decades. When really the microbiome field took off, you can think of this sort of the technology trigger in this Gartner hype cycle. A lot of people were interested about the microbiome. Is this new wave, this new technology, this new thing that's coming out, so what can we do about it? And so there was this huge peak of inflated expectations. People thought that microbiome was basically going to cure all diseases. Then people started realizing that actually the microbiome is more complex than that. And some of these results started being questioned. We started realizing how sensitive some of these techniques were. And we really started to reach this point of this trough of disillusionment where people started to think, oh, maybe the microbiome is just noise. So there's actually no signal there. But now that the field has matured a bit more, we're starting to realize that with very well-controlled methods and techniques, we actually are able to find robust signals of the microbiome with different diseases. So I think we're now reaching this sort of slope of alignment where we're trying to where we're creating more realistic expectations about the microbiome and its role in health. So what I wanted to focus now for the next few slides is what do we actually know about the microbiome? When we focus on the science, when we focus on the research, what is actually telling us? And I want to start with the best example of how the microbiome actually affects our health in a positive way. And this is really the prototypical example you'll see in every microbiome lecture. And this is the gut microbiome in protection against pathogen invasion, specifically of the species known as Clostridioides difficile. Okay, so let's break this down. So essentially, in a normal, healthy state, we have a diverse microbiota. We have a huge diversity of species, usually commensals, that are protecting our microbiome. Some individuals may have a species, so this Clostridioides difficile, that is a pathogen, but in normal individuals can be found at very, very low abundances. Now, let's say there's something happens to your microbiome. For instance, you take antibiotics because you have some sort of infection. That antibiotic treatment essentially disrupts all your commensal microbiome. What happens is this creates an opportunity for this C. difficile bacteria to essentially expand and take over. This then produces a number of toxins that causes a downstream effect in inflammation, infection, etc. in the host. And what actually has been shown is that if you take an individual that has undergone this process and is now very susceptible to C. difficile infection, if you take a healthy fecal material from another donor and put it back into this individual, you essentially reverse the whole process. So you're able to cure in more than 90% of cases. And this is why even the NHS actually recommends the use of FMTs for C. difficile diarrhea. So this is really the best example we have. And all the research that is being done in all sorts of diseases is really striving to reach this point, where we actually show that the microbiome is causing a certain disease, and we can actually recover a healthy state by doing some sort of fecal transplant. Now, another thing that a lot of you are curious about are the use of probiotics. So probiotics are essentially individual bacteria that you introduce. You take them in the form of pill. They are easily sold. You can find them everywhere. And essentially, I'm sorry to say that the probiotic research so far has shown mixed evidence about their potential use. So some studies have shown a potentially weak benefit. Other studies have shown a potential detrimental effect. And I'm showing here one particular example of two studies that came out quite a few years ago now, showing two main things that were actually really important to keep in mind. So one was that probiotics actually do not colonize an individual. So when you take probiotics, they actually don't make any long-term changes to your microbiome. And then secondly, it was actually shown that people that take probiotics, after they've undergone some massive change in their microbiome, for instance, through antibiotics, the people that take probiotics actually take longer to recover the microbiome to a healthy state. So essentially, there were two big papers that actually started to cast doubt on whether probiotics were really as meaningful and as potentially beneficial as people thought. However, what I want you also to take away from is that when people talk about probiotics nowadays, they really mean a very small number of species that have been historically used as probiotics for the last decades. What we do know now is that there are promising species that potentially do have probiotic potential creating benefits to our health that haven't actually been used as probiotics because they're really hard to put in a pill and give to people and actually have a positive colonization in humans. But this is something that the field is striving towards. Another thing that a lot of people have been paying attention to is the effect that drugs, and by drugs I mean different medicines and things that we take, affect our microbiome. Obviously, we know that antibiotics have a massive effect on our body, but there are actually newer research suggesting that even things that are not antibiotics, so painkillers, antipsychotics, non-antibiotic drugs, also have quite a lot of effect in sort of diminishing the growth of different bacteria in our gut. So this paper in particular actually mentioned that about 24% of the drugs that they tested, which were a range of different drugs that are commercially available, seem to inhibit at least one gut bacterial species in our gut. And actually, the strongest class of drugs that have a significant effect were antipsychotics. Why? I think the jury is still out there, but it does suggest that potentially there is a link of this gut-brain axis that I talked earlier about this potential effect on the gut microbiome composition. And this is really what I think cutting-edge research really is trying to understand a bit better now, is what impact the gut microbiome has in other parts of our body. And here I'm showing sort of this example of how, for instance, the gut can have a bidirectional relationship with brain function. And there are many different pathways I'm highlighting here where this could happen. You could have the microbiome producing certain metabolites, things like short-chain fatty acids or neurotransmitters that can directly affect the brain function, or it could affect sort of the gut barrier or gut function, which in itself would have repercussions in terms of brain function. So there is definitely a relationship between the microbiome and different parts of our body that we're really trying to understand a bit better, what implications that has to overall health. Okay, so this is basically some of the highlights of what we know in terms of the microbiome in recent research. But I wanted to focus a bit more now on what we're actually doing in my lab here in Cambridge. So for the purpose of this part of the talk, I just wanted to sort of introduce a bit the ways we can study the gut microbiome. So let's say we want to analyze the gut microbiome of individual. The most obvious place to start would be a fecal sample. So that's the most straightforward way of doing it. And there are essentially two main ways you can go about it. So you can take an experimental approach where you take the fecal sample, you plate it on a petri dish with a specific culture media, and you try to grow the individual bacteria that is found in these samples. This is really good because it essentially allows you the opportunity to perform experiments with those species and be able to test what they're able to do. Another approach, I'll get into more detail, is using what is called DNA sequencing. So essentially, you're looking at the genes, you're looking at the DNA of the individual organisms that are there, and trying to predict from the DNA what they're able to do and what are the species that are there. And this is essentially where my team specializes. The advantages of this approach is that it allows us to theoretically capture essentially the majority of the microbiome. We are not necessarily as constrained about the ability of certain species to be able to grow or not in the lab. And really, DNA sequencing really has, as I mentioned before, has become a staple in microbiome research. So when these technologies became much cheaper in the early 2000s, mid-2000s, so you can see here a reduction in costs, there was a massive explosion in the number of microbiome studies. Because now it's very easy for someone to take a fecal sample, extract the DNA, sequence it, and try to understand what it is. So you can do this at scale and really build these huge cohorts to try to understand the relationship between the microbiome and different human conditions. So what can we actually use the DNA for? What can the DNA actually tell us? So the DNA is sort of this composition of this different alphabet of 4 letters. And given that the DNA acts as sort of a blueprint for the bacteria, it can tell us not only which species are there, so it acts as a unique fingerprint of the individual bacteria that are there. But since the DNA encodes for certain RNA and the RNA for proteins, we can actually use this sort of blueprint to predict what the bacteria are able to do. So we can use them both for identification, what bacteria are there, and for function, what are they able to do. How do we actually obtain this data? So how do we actually go from getting a fecal sample to actually getting the data to work with? So the first step is really trying to extract the DNA first. So you have the stool sample. You essentially have specific processes that allow you to extract the DNA specifically from the stool sample. You have to homogenize it. You use a series of biochemical and biophysical steps to essentially reach this point where you have the DNA in the short Eppendorf tube. And then what you do is you prepare the DNA to be sequenced, so you add certain tags and certain markers that will allow the sequencer to recognize it, and then you process it in what is called a sequencing machine. And this is just one example, but now you actually have sequencing machines that are even the size of a USB stick. So they all come in all sorts of shapes and sizes. They all have their pros and cons, but essentially they allow you to sort of digitize your DNA and take sort of a photocopy of your DNA. So the end result is actually a digital format of the DNA. So that's why my team, we're primarily specialized in the use of computational tools to analyze the microbiome, because this is sort of the form that you usually take DNA sequencing results. Now, the process, how this usually works, is that you essentially start from this amalgamation of sequences. So imagine you have a community of microorganisms here. The different circles represent the genomes, the DNA of these different bacteria. And when you actually sequence the DNA, you get these sort of short fragments. So you only actually sequence the genome in different bits and pieces. So when you actually have these bits and pieces, you sort of have to go through this process, which we call assembly and binning, to go back and reconstruct the genomes that you originally had there. And this is a huge computational burden. This is sort of like making a DNA jigsaw puzzle where you actually don't know what the end result's supposed to look like. So you don't have the actual picture, and you're trying to build these pieces and put these pieces together into what they should be. So that's why at the end, when you build this puzzle, you'll see here, they actually are not exactly the same as they were there. You have some gaps, some fragments, because you usually something gets lost in the process. But the thing is, we now have computational methods that try to optimize this process and really try to control for potential issues and errors. So basically what I wanted to get away from this is that we can start from a complex community of microorganisms that we sequence, and then we can use these computational tools to sort of predict what are all the genomes of the bacteria that are there. And these genomes, we usually refer to them as metagenome assembled genomes, or MAGs for short. So a few years ago, when I was working in my previous post at the European Bioinformatics Institute, I spent a lot of time sort of optimizing and working on these methods to really try to enrich our understanding of the sort of the catalog of species in the human microbiome. We analyzed thousands and thousands of samples across the world to really try to build the most comprehensive catalog of the human microbiome that we could. And this essentially culminated in what we call the Unified Human Gastrointestinal Genome Catalog, or UHGG for short. So essentially a database that contained nearly 300,000 genomes of bacteria from the human gut, representing about 4,500 species and more than 170 million genes. So you can see the scale of the data that we're working with. So tens of thousands of genomes and millions of genes. But ultimately, we wanted to really understand what's new so what can we actually use this data for? So we built this catalog. What does this actually tell us? And one thing that became clear from the research we were doing at the time is that there's basically a huge gap in microbiome research. So when we looked at all the species that we cataloged, we found that more than 60% of the gut species actually weren't known before. They haven't been cultured, people haven't been working with them, they don't know what they're doing. So essentially, you can see, so in this plot here, I'm showing sort of what's called a phylogenetic tree of all the human microbiome species. You can think of a phylogenetic tree as sort of a genealogy of bacteria, sort of family history, where the different tips of the tree represent different bacteria, and essentially the lines connect bacteria on how related they are to each other. But essentially what I want you to focus here is on all this green color. So all the green that is in this tree represent all these species that were unknown before. So we cataloged, we found them in these samples, but we actually had no idea what they were doing. We had no idea that they even existed. So there's sort of this hidden universe of microbes in our gut that we know very little about, this sort of uncultured community. And this is essentially what my team now is trying to tackle. So we have this hidden universe of microbes that exist in our gut, and our vision is trying to really understand what is the relevance of this hidden community for our health. So, as I mentioned before, since we work with data that is in digital format, we essentially are specialized in the use of computational tools with techniques such as machine learning, evolutionary genomics, metabolic modeling, to really try to understand the relevance of these communities and extract as much biological insights as we can from these sequencing data. So now I just wanted to focus a bit more and show you one specific example of a paper that is actually going to come out in just a few weeks about a specific bug that we found that seems to be a key mediator of health. So in this particular project, we started out by asking a very simple question. So we have all these hidden bacteria that exist. What is their clinical relevance? Among all the thousands of species that we identified, what is the most wanted list of uncultured species that we should be prioritizing for future studies? What should we be focusing on? So we took basically the initiative to analyze a huge cohort of more than 10,000 samples coming from different parts of the world. You can see here on the map the distribution of the samples and where they were coming from. And we collected samples from various different diseases. So you can see here on the bar plot the list of diseases we looked at. We looked at things like obesity, multiple sclerosis, Crohn's disease, colorectal cancer, a range of different diseases, some directly related with the gut, others related with aspects of health. We really wanted to understand what specific microbes, what specific bugs are linked to specific diseases or are actually transversal across multiple diseases. We started out, when we started the study, we started out also by just taking a general overview of the distribution, the prevalence of these uncultured bacteria. Just asking very simply, where are these uncultured bacteria found, at what prevalence, in what proportion of the population? So we looked at the distribution of a cultured bacteria across different diseases, and we found roughly that about a third of the microbiome in each sample represented this hidden bacteria. So you can think here, all of us here in this room, we probably have about a third of our microbiome represents this hidden universe that we know very little about. Okay, so this is in terms of prevalence, but then we want to understand how accurately can we use this uncultured microbiome to distinguish individuals that have a disease versus those that do not. You probably have heard about the use of machine learning and AI approaches in biomedical research. So essentially we use similar methods, so using machine learning modeling to try to see if we build a model on the microbiome, can the model tell us whether an individual has a specific disease or not? So we tested that for various different diseases, for the ones we analyzed, so you can see here. And essentially the model performance is gauged using this, what is called the area under the receiver operating curve. And it ranges from 0.5 as being almost like random model to 1.0, basically a perfect model. And you can see that the performance actually ranged quite a bit. Most diseases had a relatively good performance, hovering the more than 0.7, 0.8. With specific diseases related with the gut, such as ulcerative colitis and Crohn's disease, having pretty high performance. And there are things like multiple sclerosis and adenomas not having as much link with the microbiome. But in general, there did seem to be a strong potential of using the microbiome as a way to sort of classify whether an individual had a disease or not. We did do some comparisons to see, okay, let's say if we build a model on the uncultured microbiome, what difference does it make if we now use the other part of the microbiome that is known, so the sort of the cultured part? We did recognize that essentially the performance was relatively similar, so the delta was relatively low, which essentially suggested that the uncultured microbiome alone almost had the same level of performance as the sort of known portion, which was quite promising because the uncultured portion, as I mentioned, is only a third. So given that a third of the microbiome is as informed as the other two-thirds, was actually quite promising for us as well. Okay, so this was just generally speaking, sort of understanding whether the microbiome could classify health and disease. But we wanted to dig a bit further. So what are the specific species? Could we find specific biomarkers of health or disease? So essentially for this plot, I'm going to be showing two types of what we call biomarkers. So biomarkers, you consider them as just bacterial species. We have disease biomarkers. So these are bacterial species that are more present in an unhealthy state, so when an individual has disease. And we have the health biomarkers. So species that are present when an individual doesn't have a disease. So we looked first at disease biomarkers. We identified about 360 what we call disease species. What I'm showing here is what is called an upset plot, just showing for different diseases how many species we found associated with them and whether there was some intersection across these. So for instance, here we have 39 species that were found together both in ulcerative colitis and Crohn's disease, 7 species in 3 diseases, etc. So out of the 360 diseases we found the species, we found 26% of the species were shared across more than one disease, and 74% were unique. So it does suggest that the disease microbiome seems to be highly specific to individual diseases with limited sharing. However, in the case of health biomarkers, this was not as much the case. So we actually found that out of 380 health species, if we will, about 57% of them were shared. So when we found them associated with health in one context, they were also found associated with health in another context. So this really reminded us of this Anna Karenina principle, that essentially says all happy families are alike, and each unhappy family is unhappy in its own way. And you kind of can think about that in the case of the microbiome as well, where it seems in the case of health, it seems that there is a more generic signal, but then once something goes wrong and there's a disruption to the microbiome, things can go wrong in various different ways. Okay, so we found all these different biomarkers, but coming back to your original question, we wanted to understand what are actually the kind of most wanted top hits that we should be focusing on. So we essentially started by analyzing all the different genera of bacteria. So you can think of bacterial taxonomy in a different rank. So you have the species, and just above species, you have the genus. So we looked at genus, and essentially we calculated for each genus what proportion of species were uncultured and what proportion of species were associated with health. So ultimately, what we wanted to find were species that were in this top right corner. So had high proportion of uncultured species and high proportion of species associated with health. And there was one group that clearly stood out way above everything else. And this uncultured gen is named CAG170. So as you can deduce from its name, it's not even well recognized. Bacteria usually have a Latin name for species. All species have a Latin name. But when they haven't been cultured and haven't been biochemically characterized, people just use a placeholder code. So in this case, this genus is currently only known as CAG170 because of how little we actually know about what it's doing. But for some reason, it seems to be one of the strongest markers of health. We dug a bit further to try to understand, okay, CAG170 seems to be associated with health. What else can we actually say about it? So we had this additional analysis that essentially aggregated the 13 conditions we looked at. So CAG170 came out on top. But we also did some ecological modeling, so try to understand what individual bacteria have a more important role for the stability, for sort of maintaining the ecosystem in our gut. And the ones that I have here, so they're essentially ranked by their importance. And again, CAG170 species came out at the top. So these species seem to be more important for essentially maintaining a healthy microbiome and keeping everything working as it should. And then we had another analysis where we actually had access to a longitudinal cohort. So basically a cohort where individuals were being sampled across time. And we actually had additional information in terms of the level of disruption of their gut microbiome, something that is called dysbiosis. So when we looked at the level of dysbiosis and CAG170, we found a clear signal of negative correlation of CAG170 abundance with dysbiosis. So essentially, if an individual had a high level of dysbiosis, so their microbiome was highly disrupted, you had lower abundance of CAG170. But if the individual was healthy and had a low level of dysbiosis, CAG170 was higher. Again, suggesting there was a strong signal of CAG170 with health. Okay, so we have all this independent evidence that CAG170 seems to be important for health, but the question that remained was why? So why is CAG170 potentially important for health? So as I mentioned before, by using DNA sequences, we can not only just look at taxonomy and what they are, but can also predict function. And this is what we try to do for this specific analysis. We essentially looked at CAG170. So here I have, again, plotted a phylogeny, and CAG170 is here. And we compared CAG170 with all related bacteria that were very similar to the species. So basically, by comparing CAG170 with related species, we could more easily find what makes it different. In this phylogeny, I've also indicated other sorts of information, so whether the species was associated with health or disease, the specific genus, and whether it was cultured or not. And you can see that in the case of CAG170, most of the species are uncultured. There's only one that has been isolated so far. But essentially, coming back to this question of what makes CAG170 unique, what makes it different, we essentially did this genetic analysis to identify what genes and functions are either depleted or enriched in CAG170, and there were two main signals that came out very strongly. One was a depletion of arginine biosynthesis. So CAG170 seems to have a lack of ability to produce and synthesize arginine. But on the other hand, it has high production of vitamin B12. So it has essentially almost the full operon of B12 distributed across all the genomes that we tested here. Now, we dug a bit further. I haven't shown here because this was really a very technical in-depth analysis, but we have some additional evidence that this B12 is actually being utilized by other microbiome species that co-occur with CAG170. So the B12 is actually not being directly used by the host, by us, but it's actually being utilized by the rest of the microbiome and all the species that are found together with CAG170. So essentially, the main takeaways from this particular work is that we found that the uncultured microbiome has a very distinct profile in both health and disease, and this has potential diagnostic applications, and that the microbiome patterns that we found are largely disease-specific. So in the case of different diseases, they seem to have a very unique microbiome, and then in the case of health, there's a greater overlap between healthy controls. And ultimately, the big takeaway that we were quite surprised about it is the discovery of this new group that we call CAG170 as a potential biomarker of health with very interesting potential clinical value. So I do see the potential of the microbiome in terms of their ability to improve human health in two main ways. And this is sort of what the group is thinking about in terms of sort of the long-term vision of the field. One is thinking in terms of diagnostics. So if we know that specific species can help us discriminate health and disease, we may potentially use them as a diagnostic application and for early prevention. So things like colorectal cancer can be quite useful because if you're able to detect colorectal cancer at an early stage, you might be able to have specific treatment options that are much more effective. And the reality here is that with diagnostics, the science is tending towards there. So it is showing that there's huge potential in microbiome. So I do believe that diagnosis is more of a short-term potential rather than using the microbiome as a form of therapy. When you think of microbiome as a form of therapy and actually using specific species, probiotics, fecal transplants, this is something that the field is still not there, and we're still really trying to understand how, one, to change the microbiome, and two, what species or combination of species should we use for different diseases. So before I wrap up, one thing I'd just like to mention as well is about this new initiative that we started last year called the Gut Immunology Brain Axis Network. So essentially, this is a BBSRC-funded initiative being co-led by different people across the UK. And we essentially have created this network to essentially help build capacity and advance our understanding of the role of the gut and the role of the immune and the role of the brain in sort of our health and how they interconnect with each other. So this network is open to everyone, not only research, but to the public, because we essentially are creating lots of opportunities, not only for funding, but training, networking, dissemination, and we want people to really get involved. So our three main visions for this network is really to try to accelerate this sort of mechanistic understanding of the GIBA, to try to train and build capacity in the UK in the field, and also increase public awareness of the importance of this gut immunology brain axis. So we do have a range of activities that are being planned for the network that will run until 2029. For researchers, there will be funding opportunities. We currently have fellowships open for postdocs if people are interested. We have regular calls for flexible funds, which are essentially opportunities for people to build interdisciplinary collaborations. I'm in charge specifically of the training aspects, so we'll be organizing regular workshops, a summer school, having mobility grants to allow people to essentially get training in other fields. And we'll also be doing a big campaign for dissemination to get the public more involved. So I do encourage you to sign up. We have a website, it's easy to find, so giba-uk.org. So if you're interested in the network, do join and be part, because this is, I think, going to drive the field forward and help quite a bit. So it just leaves me really to thank all the people that have contributed to this work. So my team at the University of Cambridge, all our collaborators both here in Cambridge and across the UK, and all our funders for the support in carrying out this research. Again, thank you for the opportunity to speak here, and I'd be happy to take any questions from the audience. Thank you very much.
Well, thanks, Alex. That was absolutely terrific. Whirlwind tour. Any questions while we're doing that? I'll just ask my own. I know of a paper where the microbiome, the genomics, was investigated as a diagnostic of autism spectrum disorder. Do you know of that, or is it
it's a controversial. It's a controversial. Okay. There was a recent opinion/meta-analysis that came out recently that really aimed to cast doubt on the association of the microbiome with autism, and essentially they were claiming that there were different studies that were using inappropriate techniques, they were underpowered. So there are some doubts about this potential link with autism. But there was original research, especially even done with mouse models, that really tried to sell this idea that the microbiome is linked with things like autism. But I think. Yeah, I think it still needs some work to really clarify that signal.
Thank you.
Hello, it's my question. That was terrific. Thank you very much. I was wondering if you're identifying, or not identifying, but CAG170, are you knowing it through what proteins it is producing? How do you recognize that piece of DNA has come from that organism, and are you looking at the proteins that those pieces of DNA are coding for and how they mix in with pathways?
Yeah, that's a great question. Essentially, the process is we first have to computationally reconstruct the genome of these individual bacteria. And there are different rules, if you will, how we reconstruct these genomes. We look for consistencies in the DNA in terms of abundance patterns, in terms of their nucleotide composition, and this allows us to create what I mentioned before, these metagenome assembled genomes. But at that time, they don't have a name, they're just genomes. What we essentially have to do afterwards is take these genomic sequences and compare them with existing databases of known sequences. Usually how they work is they extract specific sequences from the genome, what are called marker genes, and they build a phylogenetic tree, sort of the history and the genealogy of the bacteria, to place it within a specific clade. So we know that this particular group belongs to this particular family that I mentioned, so this Oscillospraceae, and is related with other bacteria that were previously named Cal-170. So that's how we essentially been able to identify this group. It's through like this sort of phylogenetic evolutionary history. In terms of the function and the proteins, essentially they are predicted from the genome. Since we have the genome, we can essentially try to annotate individual genes. So look for the start codon, look for where they end, and then we have the list of proteins that we have from this Cal-170. By having this list of proteins, again, map them to database of functions and how the proteins map to certain metabolic pathways, and we can deduce, for instance, in the case of B12, out of all the genes that we expect that are responsible for encoding B12, how many do we see in Cal-170? And this is how it allowed us to say that Cal-170 does have almost a full operon responsible for B12 production.
Hello, thank you very much. I was wondering, I saw the slide with the values of the area under the rock and connecting the genomics of the microbiome with the singular diseases. And I was wondering, the diseases where the value was low, do you think that is like something connected to the data that you currently have and the model used, or it's that you think that some diseases just don't have a connection between the microbiomes and the disease itself?
It's hard to say. The truth is, when you look at the gradient of area under the curve values we got, there is a big influence of sample sizes. So for diseases like ulcerative colitis and Crohn's disease, there are numerous studies, huge sample sizes to collect from. And that obviously helps when you're building the models. The more samples you have, the better the models can build. For things like type 1 diabetes, multiple sclerosis, adenomas, the sample size are much smaller, so that will naturally also lead to worse performance. Having said that, there is definitely, based on the research that is being conducted, there does seem to be a much stronger signal for things like Crohn's disease, ulcerative colitis associated with the microbiome. So I also believe that perhaps things like multiple sclerosis don't have as much of a strong signal with the microbiome. The tricky thing with these is that all of these diseases are complex diseases. There are multiple factors, multiple risk factors involved. Some are environmental, genetic, microbiome, diet. There are many things that can happen. So I think the microbiome would have different contributions for these different diseases. And for some, maybe it will be a small effect. But I think for a lot of the ones that are in the lower, we probably need more studies and more data to sort of refine those values and actually see if they're that low or if they can be improved with more data.
Okay, I've got a very basic question here. Are we assuming the biome in the colon is the same as in the small intestine? Obviously, fecal is doing the colon. Are we missing a trick by not sampling the small intestine if we could?
That's a great point. And it's something that a lot of research has been trying to tackle in terms of understanding how representative the fecal material is of the whole microbiome. It definitely is much more representative of the lower intestinal tract and obviously closer to the end. The small intestinal is harder to sample and it's also enriched in different bacteria that are more resistant to oxygen. So obviously the small intestine has higher concentration of oxygen, which leads to different proportions of what we call anaerobes. So in the lower intestine, you have all these strict anaerobes that sometimes you don't find in the upper intestinal. The one thing that is being done in the field is trying to think about alternative ways to sample the microbiome. There are people thinking about using things like capsules that you ingest, and then they essentially sample your microbiome at different points in the intestine, depending on pH concentration. The tricky thing with those technologies so far is that they're very variable, because they can change very easily where they sample. That can lead to very heterogeneous results, even within the same individual. And they're not as scalable as just taking, a pool sample from an individual. So that's kind of the limitation of more potentially invasive approach. Even things like colonoscopy obviously would be the gold standard where you can actually go and visualize and see what's going on there. But So for doing large-scale studies that we do, stool samples at the moment are still the best proxy we can do. But it's true that they probably are a bit of the average of the microbiome composition, not necessarily representative of what's going on in specific places.
Sorry, sir. I want to say, thank you very much for the talk. It was very, very interesting. Most of the research is probably focused more on correlation versus causation. And therefore, the sensitivity of what good looks like becomes a lot higher. I mean, you made a specific point around kind of collection protocols. Mm-hmm. How is that industry changing over the last 12 months? What are you seeing in a standardized way? Because we know that the gut microbiome is also extremely dynamic. It changes during the day. It changes based on what you eat. Is there anything interesting that you're seeing as kind of a standardized protocol in order to have comparability between studies?
It is tricky because, as I mentioned, different techniques and different methods can lead to different results. I think one of the ways of trying to combat a bit this correlation versus causation are interventional studies and obviously clinical trials. And this is something that I think a lot of research is being done. And there are studies done on, for instance, testing the effect of high fiber diet on the microbiome. So you take a population, you have your group, your control group, where you just have low fiber, another high fiber, and you see what changes have the microbiome. So if you track through time and you correlate the changes that occur in the microbiome after the intervention takes place, then you can build a better picture of causality in that situation. But that's obviously very difficult and is usually underpowered because of the difficulties in tracking an individual and actually consistently having the samples. In terms of the analysis itself, I think we are beginning to reach a point where things are much more robust. Sequencing technologies have improved quite a bit. We are moving away from short-read sequencing to more long-read sequencing, which helps with the analysis and building better genomes and having a better picture of what's going on there. And also in the computational methods, lots of benchmarking papers are coming out in terms of comparing different tools and how they work and what situations they work best or work worse. So I think there's huge developments there. My feelings that the big bottleneck at the moment, and this is sort of what my research shows, is how much we still don't know in terms of all these novel bacteria, in terms of the genes themselves. So when I mentioned that we compared the genes to a database, a lot of these databases are incomplete. So I think one of the big sort of gaps in our knowledge is really understanding what are all these genes that we find, what are all these bacteria that we find, what are they actually doing to better build a better picture of the whole microbiome when we build these sort of cohorts and build these interventional studies.
Just got a couple of questions online. One is from Paul Klein. Could you explain a bit more what you mean by dysbiosis?
Yeah, I was kind of afraid of using this term because it's a bit controversial in microbiome science. Essentially, dysbiosis is a synonym for essentially disruption. It is not a well-defined term. There's not a specific composition that relates with dysbiosis, but it's essentially a catch-all term to just mean that there's some disruption of the microbiome. What usually happens, what traditionally happens in dysbiosis is a higher depletion of the microbiome, so lower diversity, enrichment of facultative anaerobes, things like E. coli or other enterobacteria are usually associated with pathogenic infections. But there's no one definition. There's no one specific composition related with dysbiosis.
Okay, thank you. And one from David Wallace. As I understand it, our genome shows signs of aging as we age. Does our metagenome, or the gut genome, show signs of aging as well with age?
There is a bit of a signal of aging. However, the science there is a bit harder to track because as we age, there are obviously comorbidities that start popping up. And so there's a bit of a confounder in terms of what is related with age versus what is related with disease. So you have things like neurodegenerative diseases such as Parkinson and Alzheimer's. The microbiome seems to be linked with those diseases. So when you take an aging population and you find a difference in the microbiome, it's still not exactly clear if that's related with disease incidence or just the aging process. What we do know is that the microbiome beyond, when we reach adult age, so let's say 18, 19, the microbiome, it is quite stable until your 60s and 70s. So there isn't a clear signal when you compare a 20-year-old microbiome versus a 50-year-old microbiome. Actually, the biggest signal is when you compare early life, so infants, newborns, and children up to around 4 years of age versus adults. That is a clear difference. Beyond that, the differences are a bit more subtle, and they, again, will be confounded by disease state and the specific conditions of the host.
Thank you. That's it? Online? So one up there? Oh, no, one down here. On the left.
You don't have to press the button, no, you don't have to. Just go on.
I noticed that in the conditions, you had ankylosing spondylitis and Crohn's and IBD more or less at the top. And whether it's just anecdotal or not, I don't know, but there does seem to be at least some people who there is some kind of familial connection, by which I mean, for instance, perhaps the father might have ankylosing spondylitis, and one of the children might have Crohn's, and so on. I say familial. Do you think it would be genetic, and do you think there's anything significant in the apparent coincidence, not of a direct genetic link of the same condition necessarily, but of one of the three at the top?
It's a good question. There is a lot of research being done on what is the effect of genetics versus environment in our microbiome composition. When people have tried to quantify it, usually genetics is a very small portion. So it's about, I have to say, 2 to 3% of the microbiome dictated by genetics. So that means when we see these signals, it's most likely to be linked with environmental factors, things like diet or just the surrounding environment of the individual. But there are specific species that are more inherited than others. We haven't really correlated those inheritable species with the signal we found there, but it is a potential signal. For certain diseases like Crohn's or ulcerative colitis, I believe it's going to be more environmental just because of the types of microbes that are there. So there's a clear signal of inflammation in the gut. So when you have a case of IBD, you have higher inflammations in the gut, and this creates sort of a snowball effect in the microbiome, where you have enrichment of species that are essentially more resistant to oxygen exposure because the inflammatory process produces higher oxygen production with reactive oxygen species, and this leads to depletion of strict anaerobes. And those that are enriched also exacerbate the inflammatory process. So there is a clear direction of environmental and sort of disease process in the gut microbiome for Crohn's. For other diseases, there might be more of a genetic link, but again, it's really hard to entangle whether the microbiome is a result of the genetics or whether the genetics is related with the disease and then the disease affects the microbiome. So trying to sort of disentangle the triangle is quite tricky. You need very well-designed cohorts to do that.
Thanks, very interesting talk. So I wanted to drill down a little bit more on the correlation versus causation point. It's very easy to jump to the conclusion that it's the gut microbiome that's affecting your health. Is there any evidence at all for any of these diseases that it's the health of the individual that causes a change of environment that changes the gut microbiome?
It's really hard. So when I mentioned my examples, I used obviously the easiest one, which was a C. difficile case, where that is a clear signal where the individual, they undergo antibiotic treatment for something that disrupts the microbiome. They have a C. difficile infection. You do a fecal transplant that recovers the microbiome. So that's a clear. It goes with the Cox pustules, where essentially you're able to recover the disease through ability to reconstruct the microbiome. For other diseases, it is definitely the science is much more uncertain there. So that's why I think we're moving towards, I think there are probably in the next year or two going to come out more convincing research about clinical trials and what those will say in terms of using specific probiotic, specific combinations of species to reduce the incidence or alleviate symptoms of disease. So I think the area where that research is more advanced is on IBD. That is definitely an area where things are becoming a bit more clear. And there are certain species that seem to help with things like ulcerative colitis. I think one of the recent clinical trials seemed to be promising in that direction. But For things like the gut-brain axis, things like multiple sclerosis, Parkinson's disease, Alzheimer's, that's definitely the science is still very much in the air on that. We need much more well-designed cohorts to be able to say that for certain.
Hi, I was interested when you were saying that probiotics are not effective at inoculating the gut, and also you were mentioning about how that it seems to be childhood that is the key time when those kind of bacteria and gut flora are being inoculated. Has any research been done as to where these bacteria actually come from in the wild? And if you're saying they're sort of picked up environmentally, maybe from food, but wherever, is there like a geographical difference? Because on your study, you had a huge number of samples from America and Europe and what have you. But is there a difference between the sort of gut flora in those different kind of continents? And has anyone looked for these sort of anaerobic bacteria in the environment? Where do they actually come from to start with?
Yeah, it's really tricky. So as I mentioned, basically we are born sterile. That's the current idea. And the first colonizers most likely that's what the research shows, most likely comes from the parents, the mother specifically, and the environment. It usually depends on the mode of birth. So if a child is born through C-section, they're more exposed to hospital bugs, so they get more opportunistic pathogens, whereas if they're born through vaginal birth, they get more from the mother. There is a huge dynamic change throughout the first few months. They sort of start stabilizing. This is probably due to diet, so they're breastfed, they're getting bottle fed. So the microbes start to stabilize a bit more. And then with the introduction of solids, that changes then even more. So I think diet is a key determinant of the first colonizers in our gut. And the microbes that our household or our parents, our mother and our father have will also dictate very massively how things look. In terms of the geographical signal, this is a huge area of research as well, and there clearly is a very strong differences between what are called westernized countries, so more urbanized communities such as North America and Europe, versus more rural communities in, for instance, Africa, South America. There clearly seems to be a big difference there, which probably relates with differences in diet, but also the environmental exposure. Whether that directly translates with health or disease, that's kind of the question, because although the rural communities have a more diverse microbiome, species are able to metabolize a wider diversity of sugars and fibers, we don't know for sure that can automatically say that they're healthier, because they also have specific diseases that we currently don't have. So there's a bit of a question mark around that and what health impacts those have. But clearly there is a big geographical signal that in studies like ours, we definitely need to control and account for.
Okay, so this is a bit of a weird question, but you said there is a big difference between individuals according to microbiomes. So I'm thinking about in rare disease case, if you try a new drug on an individual, is there any chance that you can look at the microorganisms inside, like bacteria, and see what actual effects happen on that individual, so we can help that rare disease? Thank you.
So you mean trying to introduce new species to an individual to see if that makes a change?
It's kind of like to see if the bacteria inside, for example, gut bacteria change because of the drug you add to that rare disease patient, and to therefore find a treatment for it. So it's kind of like a reflection of the drug.
Yes. Yeah, so that's sort of one of the things I mentioned about the effect of treatment and non-antibiotic drugs on the microbiome. That is something that we still don't quite know. I think what seems to definitely be the case is that the drugs we take do have an effect. Whether the health benefits you see from taking a drug is related with the change in the microbiome, I think that's kind of the question. I guess that's kind of what you're asking. Whether the change in the microbiome could also be contributing to increase the efficacy of the drug, or is just a sort of a consequence of the drug. That actually is something we don't really know. But because this discovery about the effect of drugs on the microbiome is so recent that I think people are still trying to understand what implications that has for health.
Hey, here. Super interesting work, very inspiring. I just have this fairly specific question on when you ask, when you mentioned the B12 in CAG 70 was mostly consumed by microbes and not the host. Was the analysis done by like the correlated microbes, or?
So essentially, I'm going to get a bit into details here. So essentially, we build this sort of network analysis of CAG170 with other species based on co-occurrence.
Yeah.
Then we took the species that most strongly co-occur with CAG170 based on these network analysis, and we looked at the list of enzymes where B12 acts as a cofactor.
Yeah.
So what we found is that CAG170 itself doesn't have, at least from the current literature, doesn't have any enzymes that use B12 as a cofactor. But then when we looked at the co-colonizers, we have a huge range of co-colonizers that use B12 as a cofactor for enzyme activity. So it does suggest that the production of B12 is not for the benefit of CAG170, and the B12 is also, since it's the gut microbiome, it's being produced in the large intestine, so it's not actually being absorbed by the host, or not as absorbed by the host. But given the signal of the co-colonizer, it seems to be being utilized by those. So in addition to B12, we did find other metabolites that are being consumed, but B12 was the strongest one that we found.
Cool, thanks.
One more question from Peggy. We're working Alex very hard here, so I think
it's great questions. It's good.
Yes, the question from over there, with the comment about ankylosing spondylitis and other of the diseases which came high up in your list of... Several of those, many of them, are autoimmune diseases in their own right. So have you actually looked at whether there's a link there between, the immune system generally and the microbiome, which clearly there is a huge link. Has anybody gone any further with that?
No, there is a lot of research trying to understand the impact of the microbiome and the immune system. There are even techniques where people use essentially what are called immunoglobulin precipitation techniques. So essentially you have IgA, you have these techniques where you screen the microbiome that is being actively complexed by specific immunoglobulins. So you try to identify what bacteria are being recognized by the immune system. And there's a lot of work being done in trying to identify, for instance, which bacteria are recognized by specific immunoglobulins like IgA versus IgG, which is very important in the case of IBD. So there is some research actively being worked on that. One thing I should say is also, to your point about autoimmune disease, about them having some shared physiology. We did look at the ability, and this I did not show, about the ability of the microbiome of one disease predicting another. So let's say we develop a model for ulcerative colitis. Can that model work well for another? And one of the strongest signals we found were for ulcerative colitis and Alexandre spondylitis, two that have shared pathophysiology in terms of their immune system and having like autoimmune nature. So there is potentially something there. We are not immunologists, we're more microbiologists and genomists, so it's hard for us to dig a bit further than that, but there are a lot of good people digging further into this question.
Thank you.
Well, I think we'd better let Alex have a drink of water or something.
I'm happy to answer more.
That was a stunning talk, really interesting.
Thank you.
You explained it really well. I've come away with a few ideas. So thank you very much, Alex.
Excellent, it was my pleasure.
Well, thank you in the usual way. And I'd like to invite you to Churchill Bar if you fancy a drink, but I know you have other concerns.
Excellent.
In 2 weeks' time, we've got Professor Bill O'Neill talking about advanced laser technology and laser processing of materials. I hope to see as many as possible of you here as well. Hopefully the weather will be a little bit warmer. Thank you for coming along tonight, and thank you to both our speakers, Adam and Alex, for 2 excellent talks.
Thank you.